Hello College Biology, welcome to our second video of our genetics unit, translation. Translation is the process of protein synthesis. That means we're making proteins. So let's get to it. Translation follows transcription, which means we have already created a sweet copy of a gene in the form of messenger RNA right here, and that is in the nucleus. But all of our sweet protein factory materials, ribosomes, are outside of the nucleus. So step one of translation is that the mRNA leaves the nucleus through nuclear pores. So there's more to it. Once we are outside and we have left through those nuclear channels, the mRNA attracts the small ribosomal subunit, which happens to be made out of ribosomal RNA or rRNA. Then the first amino acid comes along, and more importantly, the large ribosomal subunit will bind as it is attracted to the small ribosomal subunit, and now they will create a full ribosome around that mRNA, and we are ready to go. That mRNA is going to get pulled through that ribosome, and while that is happening, tRNA, or transfer RNA, are going to be attracted to the mRNA. Well, how does that work? Well, each mRNA has codons. These codons are a sequence of 3 nitrogenous bases that, through complementary base pairing, will attract 3 nitrogenous bases in the tRNA. So each codon will attract different tRNA, but each specific tRNA will bring with it a specific amino acid. So different tRNAs will bring different amino acids. So this tRNA has this anticodon, which will pair up to a codon and deliver serine, whereas this tRNA will bring aspartic acid, and this tRNA with a different anticodon will bring arginine. So different tRNAs bring different amino acids. Well, what does that have to do for us? Well, if we review a little bit, we've got the mRNA copy of the gene. We've got tRNA, which bring amino acids. We have the large ribosomal subunit and the small ribosomal subunit. And those things will bind together to get us started in this process. mRNA, small, large, and then our first amino acid. Now, it's here things get interesting. We actually start making some protein. The mRNA gets pulled through, the tRNA are attracted, and each tRNA is added to this growing polypeptide chain. The ribosome uses energy, ATP, to make covalent bonds between the amino acids, and it continues to create this chain. Now, something that this image isn't illustrating very well, that this image kind of does a little better job with, is that this amino acid chain, this polypeptide, as it is being made, it does fold while it is being made. It folds in the middle of the process. So as we continue to add amino acids here, we continue to see folding here. So maybe we got a beta sheet going over here, and we've got an alpha helix going over here, and all of those protein structural components that we've discussed, that's happening as the polypeptide is being made, which allows us to go back here. This continues, adding to the amino acid chain and having that polypeptide fold because of those interactions. That continues until the ribosome reaches a stop codon on the mRNA. The stop codon will bring a release factor. And the release factor, when it gets there, it will isomerize and it'll disrupt the hydrogen bonds that are kind of bringing everything together. And when it does that, things start to dissociate. The completed polypeptide is released, and we kind of separate all of the components we started with. So the large and small subunits of the ribosome are separated, and the mRNA is released, and each of these parts could be used again when another transcription unit is sent out from the nucleus. So let us review. Translation is the process of protein synthesis, and it starts with when mRNA leaves the nucleus through a channel. The small rRNA or ribosomal subunit of the ribosome is attracted to the mRNA, and the large protein subunit of the ribosome is attracted to the small subunit, forming a complete ribosome around the mRNA. The mRNA is pulled through the ribosome, and its codons, which are 3 nitrogenous bases, attract specific tRNA, which is transfer RNA, through complementary base pairing. Apparently, I spelled that wrong. Okay. Each of those tRNA are significant because they each bring a specific amino acid to the ribosome. And using ATP, the ribosome attaches the amino acids together to make a polypeptide. And as that is happening, the polypeptide folds, and this continues until the ribosome reaches a stop codon, and the stop codon attracts a release factor, which triggers the dissociation of the two ribosomal subunits, the mRNA and the protein. I hope this helps. Struggle well.